Hello, this is Professor Gerada. Today in lab, we will be talking about molecular genetics, specifically the techniques of PCR, restriction analysis, and electrophoresis. Let's begin with a review of DNA structure. As you'll recall, DNA is made up of two strands of nucleotides. Each nucleotide has three parts. A nucleotide is composed of a phosphate group, which contains two negatively charged oxygen atoms, a deoxyribose sugar, and a nitrogenous base, either adenine, thymine, guanine, or cytosine. Each nucleotide is connected to the next nucleotide by a covalent bond called a phosphodiester bond. This is a very strong bond. The two strands are connected through hydrogen base pairing between two nitrogenous bases. So here we can see two different hydrogen bonds between adenine and thymine base pairs, whereas guanine and cytosine base pairs have three different hydrogen bonds. Now let's discuss the central dogma of molecular biology. As you know, the DNA we were just talking about carries the hereditary material of all living things. That DNA can get copied into other DNA molecules just before cell division in a process known as replication. The DNA can also get copied into a similar nucleic acid known as mRNA. This is called transcription. And finally, that mRNA can get converted into protein in the process called translation. And it's the protein that actually does the work in the cells. And these proteins can look very different depending on the DNA sequence or the gene that they originally came from. So in this way, we get from a DNA sequence, basically known as the genotype, into the phenotype, which is what the cell looks like or what the cell does, which is determined by the protein. In this lab, we will be investigating the central dogma by looking at the human HBB gene. The human HBB gene comes in two different varieties or alleles. The HB8 is the wild type allele. At a particular sequence in the gene, we see GAG. Now remember that each gene is thousands of nucleotides long, so this is just in one small area. In some individuals, there's a change in the DNA sequence, a mutation, which reads GTG. So the HBA allele is the wild type allele or the normal allele, and the mutation is called the HBS allele. So this gene gets transcribed into mRNA, and as you should remember, the non-template strand, in this case the top strand, looks just like the mRNA except that T's get converted to U's. And that mRNA will be translated into a protein. So the human HBB gene codes for a protein known as beta globin. So beta globin makes up part of your very important molecule called hemoglobin, which you probably already recognize is the protein in your red blood cells that carries oxygen. So in the normal or wild type version of beta globin, we have a glutamic acid in a certain position. Glutamic acid is a charged amino acid. The mutant allele, the HBS allele, codes for a valine amino acid at that same position. So the valine ends up on the outside of the hemoglobin molecule, and it can clump together through hydrophobic interactions. This causes great problems for the cell, and in fact it is responsible for the sickle shape we see in individuals with sickle cell disease or sickle cell anemia. What happens is that these abnormal sickle-shaped cells can clog up the capillaries leading to tissues and organs, preventing blood flow and causing great problems for that individual. So remember that in each of your cells you have two copies of every gene. So here's a plate of spaghetti, if you will, that looks like the DNA in an interphase cell. And so The little colors here represent our different alleles, our two different alleles that each one of us carries for the HBB gene. So remember that we have about 20,000 different genes in each one of our cells. So the HBB gene is just the tiniest part of that. So you could have two wild type alleles for HBB. So each one of those alleles make normal beta globin protein. Here's the second allele somewhere else in the DNA. And so this person's genotype would be, remember the genotype refers to the alleles carried by that individual, so they would be HBA, HBA, two normal alleles. And their phenotype, what they look like, would be normal, or kind of a nicer way to put that would be unaffected when we're talking about genetic diseases. A person could also have two mutant HBS alleles in their cells, so they would make entirely the sickle cell version of the beta globin protein. Their genotype would be homozygous for HBS, so HBS, HBS. And their phenotype would be sickle cell disease, the severe form of the disease where blood flow gets blocked to major organs in sickle cell crisis. And finally, a person could have one of each of the alleles, so they would make wild type protein, normal beta globin protein, and also the sickle cell version of the beta globin protein. So their genotype would be heterozygous, so HBA, HBS. And the phenotype is what's known as sickle cell trait. So in heterozygous individuals, only under extremely poor oxygen conditions do they show some symptoms of sickle cell disease. So our job in this lab is to genotype and diagnose the individuals of the Ryan family, a fictitious family with a dad, a mom, a little girl, and a baby boy. So how can we do this? So we're going to do this in 3 steps using our biotechnology techniques. First, we need to isolate and amplify part of the HBB gene surrounding this mutation. We're going to do that using the technique known as PCR, using HBB gene-specific primers to get that HBB gene out of that whole plate of spaghetti. Next, we are going to cut that PCR product with an enzyme called DDE1, which is a restriction enzyme that specifically and only cuts the HBA allele sequence and not the HBS mutant allele sequence. And lastly, we need to visualize our DNA and interpret the results using DNA agarose gel electrophoresis. We're going to let Paul Anderson from Bozeman Science help explain these 3 techniques. So over to Paul now to introduce PCR.
This was invented by Kerry Mullis. He was riding his motorcycle one day and came up with this idea. And so to do that, we need a few extra things in our PCR machine. One thing we need is a primer. Primer is going to be a little section of DNA that'll grab onto the DNA. It'll allow that polymerase to drive down the DNA. Now, where's Taq polymerase from? Remember, it's that bacteria. Because in the PCR, we're going to heat this up really hot. What else do we need? We need nucleotides, so we need new letters. And so let's check this out. Basically, we put it in a PCR machine, which looks kind of like a photocopier. You put your DNA in the top, and then it's going to quickly heat it and cool it and heat it and cool it. And so as it heats this, it's going to unzip that DNA in the middle. It's going to break all those hydrogen bonds. What's the next thing that's added? Well, the next thing that will be added is going to be the primer. The primer is going to bond to the complementary sides of the DNA. Now, we create the primer because it's going to target a specific gene that we want. What happens next? Well, once the primer's in place, then Taq polymerase is going to grab on. Now, this looks familiar. If you know anything about DNA replication, how does that work? Well, we unzip our DNA. It's helicase that does that. We put a primer down, and then it's going to be DNA polymerase inside us that makes copies of our DNA. But in a PCR machine, it's Taq polymerase, and the reason why is Taq polymerase can withstand these really hot temperatures. Okay, watch carefully what happens next. Well, basically, as the Taq polymerase runs in either direction, it's going to add complementary letters to either side. And so basically what do we have? We had 1 strand of DNA, now we have 2. So the PCR machine will cycle. It'll again heat up the DNA, so it unzips those hydrogen bonds. We're going to add primer on either side. Taq polymerase is going to grab on, and as it races down, we're going to produce complementary sides. And again, heat it up. We're going to unzip the sides. The primer is added on, Taq polymerase on, and now we have, if you look at it, we've now got 8 strands of DNA. And so this just in a few minutes, we've gone from 1 to 2 to 4 to 8, and now we just get 16. You get that.
Okay, so for our particular PCR reaction, we're going to use primers that amplify that section of the HBB gene that surrounds that mutation site. Okay, so we have primers on either side of that position, and what's going to happen is that we're going to amplify, make many, many copies of the portion of the gene surrounding that sequence. So, depending on the genotype of the person, we might be amplifying the wild type allele, or we might be amplifying the HBS allele, or if they're heterozygote, you're going to be amplifying both at the same time. Now that we've amplified our portion of the HBB gene using PCR, we're going to cut our PCR products with a special enzyme called DDE1. We're going to have Paul Anderson introduce to us the concept of restriction enzymes.
Where do restriction enzymes come from? Well, basically they come from bacteria, because bacteria have been locked in this million-year war with viruses. And the viruses, you can see, inject their DNA into the bacteria DNA, and they make more viruses. And so how do you fight back? Well, basically what they do is they methylate their DNA. First of all, what does methylate mean? You basically add a methyl group to all of their DNA from the bacteria to protect it, and then they're going to secrete these or create these restriction enzymes. And what the enzymes do is they chop up or cut DNA. Since all of the bacterial DNA is protected by these methyl groups, what it's really going to chop up is all this foreign DNA. And so all of that viral DNA is broken apart, and then the bacteria, after it's done that, can return to its specific shape. So let me show you how a restriction enzyme works. So if this is a section of DNA, a real common restriction enzyme is something called EcoR1. It comes from the word E. coli. This is a restriction enzyme 1. And basically what it'll do is it'll scan the DNA until it finds this specific sequence, and it's literally going to cut it in half. And so if I were to find that, where is that going to be up here? Well, you can see we have a GAAT, so it's going to cut right like that through here. It's going to go all the way to the end, and it's going to cut it in half like that. And so if we take a look at that, basically what will it do? It will break that into 2 fragments. If we take another one, oh, well, did you see that? It came right back together again. So what brings it back together again? There are going to be little hydrogen bonds here between those 2 ends. And in fact, we call this a sticky end and this a sticky end because there are going to be hydrogen bonds formed between the 2. And so once the enzyme gone, it comes right back together.
So in our experiment, we'll be using a restriction enzyme called DDE1. So our restriction site for DDE1 is CTN, where N could be any nucleotide, and then AG. And so this enzyme will cut that sequence, if it exists, into 2 different pieces. And so how are we going to use this to figure out the genotypes of our family? Well, it turns out that if you look at the HBA allele, the wild type allele, here's that GAG, which changes to a GTG in the mutant S allele. And you'll notice the 2 nucleotides just to the left of that are C and T. So in the wild type allele, we have a perfect site for DDE1 digestion, whereas that mutation changes the sequence, and it is no longer a DDE1 recognition site. So if we try to cut our PCR products with the wild type and the mutant versions of this gene with DDE1, what happens is that the HBA allele gets cut because it has the correct sequence, and the HBS allele remains uncut. So we get 2 pieces of DNA for the wild type allele and no cut, or just 1 piece of DNA for the mutant allele. This type of scenario is usually referred to as a restriction fragment length polymorphism, or RFLP. And that refers to the situation where, depending on the allele, you get different size fragments of DNA after you digest with a certain enzyme. Okay, so where are we? First, we've PCR'd out the portion of the HBB gene surrounding that mutation site. We have cut our PCR products with DDE1, and what's next? So, at this point, our DNA is invisible to us, just a clear liquid in a tube, and what we need to do is to visualize that DNA and then interpret our results to determine the genotypes of our family members. So once again, back to Paul Anderson to introduce to us the topic of DNA agarose gel electrophoresis.
Now we have to figure out how to separate the DNA according to its length. And so to do that, let me talk about pachinko machines. Pachinko machines are essentially these vertical machines. They're really popular in Japan. You put a ball up at the top. You usually flick the ball up at the top. And then that ball is going to bounce down. And if it eventually goes where you want it to go, if it eventually goes into a little cup down here at the bottom, then you might get more of those balls back. But in that case, it wouldn't. So imagine a game of pachinko where we had one ball like this that's bouncing its way down. And then we had, instead of another ball, we had like 8 balls that are attached together. So something like that. So what would happen to them if we dropped them in a pachinko machine? Does it make sense that they're sometimes going to go slower, they're going to wrap around, it's going to take them way longer to get down to the bottom. And so the single pachinko ball would already be at the bottom where this big chain of pachinko balls hasn't even made it very far. And so how does that apply in DNA? Well, think of DNA instead of pachinko. So basically, if we were to take a small fragment of DNA, it's going to work its way farther down through that pachinko machine sooner than one of these big fragments. And again, we don't use pachinko machines, but we do use gel electrophoresis. And so how does this work? Basically, inside you have a gel. The gel is kind of the consistency of Jell-O. It's going to have little wells on one side where you insert the DNA. And what's pulling the DNA? Well, in a pachinko machine, it's going to be the gravity. But here you can see that there is a red cable. That means there's a positive charge on this end. So there's a positive electrode all the way across. There's going to be a negative electrode. You can see it right back here on this side. So basically, the DNA is going to migrate across that gel. DNA has a negative charge, so it's going to be pulled towards the positive end. And so if we look at this, this is kind of that, if you've ever heard of like a DNA fingerprint, what's going on? Well, basically the DNA is going to be pulled in this direction. We could say this, since it's been on for a while, that this fragment right here is going to be bigger than this fragment right here, because this small fragment has made its way farther from these original wells where it was up here. And lots of times you'll run a ladder as well. A ladder is a bunch of DNA with known distances or known quantities, and so you can read across and see how big is that, how many nucleotide pairs do we have. When we do this in class, we use a dye called ethidium bromide, and basically it'll dye the DNA. You can put it under black light and you can see where the DNA is.
Okay, so here are the results of our gel. In each separate lane, we have the PCR and restriction digests from our family members, the father, the mother, the daughter, and the son. In the first lane, we have our DNA marker, which we use to compare the size of our DNA products to. So what do we have here? What is this DNA fingerprint, and what does it mean? Recall this simple map of the HBA allele, the normal allele. These are our PCR primers, which determine the ends, and the DDE1 restriction site. So after we have PCR'd and digested our products with DDE1, we have 2 pieces of DNA coming from this normal allele, the smaller one and the larger one. The smaller one corresponds to this band on the gel, and this band corresponds to the larger piece of DNA. Now recall that the sickle cell or mutant allele, HBS, has no DDE1 restriction site because of this mutation. So we are just left with a single fragment of DNA, our original PCR product. And that's this larger band that we see on the gel. I'm going to leave it up to you then to figure out the genotypes and phenotypes, or diagnosis, of each of the family members based on your gel results.